Amy, could I ask you to join me up here and Cheryl, please? Thank you very much. Great presentations, both. But before we get to questions from the audience, I wanted to see if you'd be willing to blue sky with me a little bit. So I give this example of what happened in acute lymphoblastic leukemia in children. So what would have to be in place? Actually, so I'll frame it for you. If the pharmaceutical industry had populated the medicine chest with powerful targeted agents, and I think they're well along the way of having done that, and if the kind of molecular studies that Dr. Trent and Dr. Breen and others talked about today were in place, and if you imagined you wanted to dramatically change the standard of care for sarcoma in dogs, what would have to be in place? What would we have to do to be able to make that possible? Have thoughts about that? Sorry to put you on the spot.
Yeah,
I just...
I mean, I think we have a pseudo infrastructure in place to be able to do that. You know, it's twofold. One is to get oncologists on board with getting animals into clinical trials. I mean, I think hands down, we as a community have to say we're committed to this, we want to do it, we're willing to offer it up front to our patients, even if it means in private practice that we may not be getting as much income, but that this is an important mission. And, you know, I think from all of our standpoints, I've been doing this for a really long time, not 200 years, as Doug might think I have. But you know, I don't want to do the same thing over and over again. I don't think anybody does and see the same failure. And so we have to all be engaged as a community. We also have to be given the right tools to do those studies correctly. And that means training with respect to GCP, with how to collect specimens, and to have the facilities to do so. I think it's doable. I think if people really get on board, there's certainly enough cancer out there. There's enough specialists now spread throughout the country that we can do it. But it's going to take an infrastructure, organization, and training.
Speak. You don't press the mute button. Okay, speak button. Thank you. I would echo the training aspect, because I think that when you look at the oncology residents that come out of training programs now, the vast majority of them go into private practice positions, and they're coming out with this amazing skill set, and we haven't done a good enough job of, I think, maybe offering, and we need funding to support training programs. But then also, I think, getting to veterinary students really early on in their career and telling them, you know, you are about to have 4 years of training in a specialty that makes you extremely unique and very knowledgeable about comparative medicine, not just oncology, not just surgery, not just giving vaccines, not just doing whatever. You have the chance to really make an impact as a scientist, and they don't see that. And so if we had the opportunity to really support them and see more of them enter into the academic world, I think it would be better too, because we would have more people heavily engaged in the enterprise.
Thank you. Questions from the audience?
Yeah, Michael Larimore, UC Davis. Cheryl, maybe you could amplify a little bit or explain what you made a comment about, that some of the outside practices will go for standard of care, and that you encouraged, or sometimes what would happen is they would go right onto the trial at OSU. Could you clarify that a little bit? Because this will come up tomorrow in our opening session about some of the tricky parts about the ethical aspects of that. Could you comment further on that?
So for many of our clinical trials, when patients are referred in, they've already sort of burned through standards of care. So that would be, you know, for lymphoma, CHOP, and maybe a rescue regimen or two. When a patient comes into Ohio State and they're evaluated and diagnosed, the owner is given upfront all of the options. They get to choose. It's completely up to them. But we say, this is what we can offer you with respect to standard of care. This is what's known. And then there's these 2 clinical trials or 3 clinical trials, and you can read these informed consents and decide, is this right for you? Is this something you're interested in? And then if they're interested in it, then they start to interface with the clinical trials office to give them a little bit more information. And they may, after they hear all of it, decide not to do it, decide to wait and make a decision. But that's been offered upfront. So it's not a situation where a number of therapies have been instituted and then the decision is made, well, I've run out of options for you, so I'm going to see if I can send you for a clinical trial somewhere. Because one of the advantages we've talked about is the fact that we really aren't sort of hindered or burdened or required to engage in a standard of care. And the huge advantage we have is being able to engage in some of these novel therapies in the setting of naive disease, which can often provide really critical information that you don't get when a patient has life-limiting comorbidities. So that's sort of the difference in sort of the mindset.
Great.
Wendy Shelton. Amy, this is for you about the confidentiality. You know, when you have a sponsored trial and you kill a drug or you discover something about it, it's not going to go forward, the sponsor's not going to want that to be public. And at the same time, we need case studies. This is really a lot about validating the model. So how does that fit into what you do?
So our agreements with our sponsors certainly have time limits in them as far as how data is held in confidence, and that there is a joint decision on when to publish and where, and sort of a back and forth agreement about how the data is managed. I think it's something else that we're always sort of really aiming for is to get our data out in the literature. And has that happened, obviously, with every one of our COTC trials? No. I mean, you've heard already we have 5 published manuscripts. One will be out shortly, but it's in press, one of the 5. So, that's always a goal. But we understand that there are strategic decisions behind drug development, and that it may not get us published as quickly as we'd want it to. But that's always a goal, that if maybe we don't even publish the entire body of work, we can at least break a piece out and get it out there.
It just feels like it limits the connections. Cheryl, do you have the same situation?
So there are instances where we've worked with companies and ended up killing a drug. And those have not gone forward through an IND process. And so there's really no reason to publish it. And so we, with the sponsor, have come to agreement that this is not something that we're going to publish, mainly because they didn't move it forward beyond some of the mouse work.
And yet that kind of validates what we're trying to do, because killing a drug early is a good thing.
Right, but we also have to respect the needs of the sponsor to some degree. And, unfortunately, particularly for the smaller pharmaceutical companies, they're dependent upon continual investment. And that's really important for their pipeline to continue. And so for them, it's certainly something we could potentially revisit later on, but we're cognizant of their needs and the risk they take in working with us.
Let's move over here.
Thank you. Rowan Milner, University of Florida. Question for Amy. Thinking of the 30,000-foot view of all of this, we've reached the point in the COTC where I think you've reached the critical mass, that the word's out there. What about the COTC or related organization that you're part of becoming a study section that would offer RFAs specifically for this? Because we know that DVM PhDs are really pretty much cut out of NIH funding and less linked to medical schools or so on. And I think that this might be a good driver for specific research. And maybe to answer something that the previous person had was, if you're driven by pharma, then you're not going to get all the information out. If you're driven by RFAs, maybe that would be a better option.
Thank you for that. Yeah, I mean, I think that, are we driven by pharma? Not necessarily. I mean, I would say that a component of the studies that we have executed and are executing on currently and plan to execute on, component of them are pharma driven, but not all of them. I mean, what you're suggesting would be that we move to the extramural NCI and we become a part of that machinery. I mean, I think that carries with it, benefits and risks. Every sort of, place where we would sit would carry those benefits and risks. So I guess I'm not prepared necessarily to comment on that currently.
Yeah, thank you, Amy.
Yeah.
I'm surprised the funding question didn't come to me. Please.
I actually had one, sorry, Tanya Zapka from Genentech. I had one response also to Wendy. A lot of the data is not useful publication information if you don't understand mechanism on or off target, if you can't explain the findings. So then it just becomes kind of nonsensical literature. But I imagine when you guys go back and like we do incidents of success across pharma, and it'll start going into these Gantt charts and where you have success and, efficacy or PK or safety. And so always publishing the results are not always useful if you don't know what they mean and you can't put them into context. So that's just kind of one comment. I think another comment is that a lot of clinical trial data is starting to be shared, and companies are opening up their clinical trial databases because they're realizing that sharing that data is better than keeping it close to the chest. So I think that landscape is also changing. And then I think just one other comment, because we were looking for gaps and opportunities, and I guess I'll go back to safety because that's where I live in the pharma world. But I think the COTC, and if folks aren't aware of this, because I don't know everyone's background, but there are two large consortia that work on new safety biomarkers, translational from preclinical to clinical. And since you're running these trials, and I don't know who all your sponsors are, I know AZ, Gilead will know these, but some of the smaller companies might not be aware of new monitoring tools, and certainly know that we look at them in healthies, but we don't really know much what they do in patients. And so it might be an opportunity to reach out and start helping build that database as well. And the consortia may be helping to pay for some of that work as well. So it might be another opportunity, especially in kidney, liver, vascular. Those are some big ones.
Thank you. Please.
So this is a comment to something you said, Lou, and I can't help myself because I'm a pediatric oncologist and I don't want people to walk out of this room with the wrong impression. So if you look at ALL and you say 85% of our patients are surviving, it's a fantastic success. The longer our patients survive, the more we understand the horrendous toxicities they pay long term. And I will tell you, having been in this field since 1983, every year at ASCO, our data looks worse and worse and worse the longer our kids live. And I suspect as we cure more patients with targeted therapies, hopefully we will learn that there are toxicities that are paid down the road. Now, hopefully our targeted therapies won't be as toxic, but this is a never-ending problem. And so, I don't want people to walk out of the room feeling like we've solved the problem of ALL, because our children that we cured when they were 4, who are now 40 and 50, have horrendous problems that they are paying the price of that we weren't aware of 35 years ago. So I just didn't want people to walk out with the wrong impression.
Thanks for clarifying that. It's actually a substantial effort we have underway. Clearly, the collateral damage that comes along with high-dose chemotherapy is a problem. We have to fix that. Please.
Yes, Michael Sagaler, Conafe Group. Quick question for Cheryl, very quick question for Lou. When we were working with CSU to put this together, it was very important to get the voice of the patient here, so we appreciate Lou being here. So the brief question for Lou is, what do you feel the awareness of comparative oncology is in the patient community, and how can we increase that? Because they are, in a very good way, a very driving force and could help push things along. And then the very brief question for Cheryl is also, we're looking for more sponsored trials, and the examples that you gave with Karyopharm is wonderful. And how do you see from that relationship with Karyopharm, is there lessons learned that could be used by others to increase the number of such trials?
So I have a very quick answer for you. I suspect the awareness is low. I'll be honest with you. I'll put it in the context of the Leukemia and Lymphoma Society. Last year, we jumped a major hurdle. We've now deployed over $1 billion to support research. And the community doesn't know about that. So I suspect that they won't know about the comparative oncology efforts either. And I think we ought to join together to figure out how to fix that problem. Cheryl?
So with respect to knowledge in the community, it certainly is a hurdle. And we have realized that we've tried to reach out to veterinarians to get them to refer cases in and to make awareness of our studies. And it hasn't worked very well. And I think it's not purposeful. I think they're just very busy in their daily lives, and it's hard to remember that there's clinical trials out there. So we've sort of taken a different tack, which is to just reach out to the public and engaged in a massive sort of marketing campaign within Ohio State and the Columbus area. And this is through media, so radio and TV. We have, like in the campus area, buses. There's all sorts of signage about clinical trials. And so, and Facebook and Twitter and all those sort of media connections that you can use to reach out to the public. And it's paid off because now it's the people that are calling, not the ref vets. So we're getting clients calling about getting their dog in, and they've heard about us. And so I think the more that they hear and the more that they know, the more that they tell people, et cetera. And that makes there's sort of this ripple effect, and it makes a really big difference, and it's starting to pay off. With respect to the Farm work, this was, a lot of these connections happened. You knew somebody from a while ago who knew somebody else who, decided to give you a call. And it's been a great partnership that's ongoing. Often with these companies, when they learn something that benefits them, when they've moved into human clinical trials, they come back to you with their next drug. Which has happened with both Cinta and Farm. And really, from the standpoint of, particularly of small companies, their risk benefit is really different than with big companies because they have less capital to work with and they need to show proof of clinical benefit in order to keep moving forward and to fund subsequent studies. And so from the standpoint of the ability to raise funds, they're seeing proof of principle in a spontaneous tumor model is really leverages them within the investment community. And so often, particularly at AACR, we'll get some of the largest traffic comes from analysts that are looking at our data and then it gets posted online and then, so it positively impacts in that manner. I don't know if you had anything to add.
Yes, I just actually want to come back to, Lou, your first question and point on the blue sky and sort of reflect on the current blue sky. And so the first point I'll make is, again, Dr. Kasten stealing slides of dogs looking like humans.
Mm-hmm.
I mean, that's progress. That's a reflection of the blue sky. In this last year, we have a major pharmaceutical company in the U.S. developing their own internal comparative oncology program. That's a blue sky.
Mm-hmm.
So I think that we have a cooperative group within the U.S. endorsing the use of the dog as a means to define their next upfront clinical study. That's another blue sky. And if I imagine forward, I'll sort of respond to Dr. Barrett's question of me. And so, what are we going to do, or should we develop a mechanism to prospectively credential the targets in canine cancers so that people like Carl can go through a list and say, that's what I should spend my money on? And so I asked the question. I think the answer is yes. I'm interested in your thoughts. And I do think, and Cheryl and I have talked about this, there's value in the prospective development of that data through integrating these sort of questions in clinical trials. But I think in parallel, asking those broader biological questions of tissues, as Dr. Breen indicated, already exist, seems like a pressing opportunity.
What is, we need a TCGA, yes. Yeah, I would say the CCAGC offers a really awesome opportunity to think about the blue sky component of cancer genomics in dogs. And those samples have been collected, they are there, they are ready to go. So I think that would be an amazing initiative for us to undertake as a community to make use of those samples that are of extremely high quality, who have clinically annotated data from the patients, they're treatment naive, and they're from relevant histologies. And that whole effort to get CCOGC moving represents a really nice roadmap for other prospective tumor banking initiatives.
Okay. And I'll just quickly return to a point that Dan made and that Jaime, you and I shared in one of our discussions, and that is that, so it's great that we identified these targets, and let's say we do it and we have these targets and we can go down the list, but it is true that credentialing those targets with biology in the dog is also going to be necessary for us to really answer Carl's question. So I think there's two components to that.
So let's see if Carl has another question.
Yeah, my question relates to 2 points you made in your presentations. One about standard of care and the other about exceptional responders. So having this database, having these tissues and their medical records, do you have clear information to provide owners as to what the expected outcome will be under standard of care for their patients? And then secondly, what do you mean by exceptional responder in the canine cancer world? I mean, how are we talking, a year? What would be an exceptional responder?
So if I understood the question correctly about giving an owner an expectation about what regarding the standard of care would provide for them. So with some diseases, we have a pretty clear understanding of what the expected outcome is with a given patient or a group of patients that have, you know, diffuse large B-cell lymphoma or osteosarcoma. And so we can tell the owner, this is the typical way we treat, this is the typical outcome. You know, you can fall on either side of the curve, but most dogs live X number of months with this treatment, and this is what we expect to see. There are some disease entities where we don't have this as clearly, but for many of them, there's a large body of published data. I mean, most of us have been using these same therapies for 30 years now, and there's a published body of data. And they're not in the same manner that human clinical trials have been accumulated, but we have a pretty good sense. So owners really understand that this is what they're going to get for what they are going to do for standard of care. And remember that for most of these people, this is a cash for service business. So they're paying out of pocket. There's nobody else paying for this. So the consumer is directly able to choose what path they want. What was the second question? Sorry, my age.
What would be the typical
Exceptional responder. So in a
typical duration for a dog with lymphoma?
so
For those who don't know.
So it depends on what the question is that you're asking. For our PI3 kinase inhibitor study.
CHOP.
Oh, for CHOP, for an exceptional responder for CHOP, we would say probably 2 years. We have dogs that live 5 years, that stay in remission for 5 years, and those are exceptional responders.
It's like less than 1% though,
Yeah.
probably.
Yeah, I mean, so, but
Yeah.
that's not, I mean, again, what do you tell, you tell your patients take CHOP, you'll get 5 years? Well,
No, we tell them no, that's not what we say. We would be lying. We tell them like,
Yeah.
You treat lymphoma, and you don't really know what it is. And so the exceptional responders may be dogs that we're going to do well if you send them home on pet candy. And
right.
you might actually kill them if you treat them.
yeah, so you're making my point, which I was trying to get at. It's a lot of anecdotal information. So to me, having good data through, all of you have it, collating it and
Right.
presenting it well would serve you well. And again, of course, I would define exceptional responders.
Right.
In that context.
Thank you. I want to move on because we're running over
Why
time.
would the
We've got-
NCI not do exceptional?
We have until 5:15.
for the
We have until 5:15.
Why would the NCI not do, NCI has an exceptional responder, where are you, Lee? Why would they include dog tumors in addition to the human tumors?
Why would they or why wouldn't they? Why would they not look at dog tumors as, well, I mean, I think if you have issues drilling down on what somebody believes is a good response. So, you know, a lot of people are like, well, a year, what's a year? Well, a year to a dog is like 5 to 7 years to a human patient. I mean, I hate to do that whole dog year thing, but truly, I mean, and then it's not just about duration of response. You could also think about the response itself. If it's a complete and utter disappearance of disease promptly within, you know, let's just say 7 to 14 days, you could also classify that as its own exceptional response. So I think the devil is in the details here.
Be careful about that.
Okay,
I mean, I've seen lots of complete responders who come back
sure.
roaring.
So, but then that, you've just pointed out there's an opportunity there, and that you look at pre and post baseline and then a relapse.
If you did the exceptional, really truly exceptional responders and the dog, that would tell me is an opportunity exists to go after that tumor.
Sure.
Yeah.
Yeah. If you have the adequate samples banked ahead of and after the disease recurs, that's the key, is having the adequate samples for analysis.
All right.
So Michael Caston, Duke. First, I have to respond to Dr. Khanna's comment. It's not so blue sky that I feel an affinity to veterinarians. As a pediatric oncologist, we share a lot in common. Not only do we treat the same tumors, but we have to use our diagnostic skills to diagnose the patient. They can't tell us where it hurts or what the problems are. So there are a lot of similarities. So my question is for Dr. LeBlanc, and maybe it's similar to what Dr. Barrett was discussing about opportunities within the comparative oncology program at the NCI. I think a recurring theme that we heard today is one of the clear gaps in the canine oncology community is really full genomic understanding of the mutations that are in the various tumor types. And Dr. Breen addressed, has very elegant work with the chromosomal abnormalities, but I think what pharma is going to require from us, especially in the era of targeted therapies, is really well-characterized mutational spectra, whether or not it's the driver mutation, just to know that for doing the kinds of trials. So though it's not within COTC, within the comparative oncology program, there was a genomics activity that you listed within the comparative oncology program within NCI. Can you tell us what that genomics consortium is doing and whether that's something we could take advantage of to do more formal genomics analysis of the canine tumors?
So it's the CCOGC biospecimen repository that our program, through the generosity of the CCR, helps us maintain not only the executive day-to-day management of that through our program, but also keeping literally the freezers and the lights on. Because all those specimens are there's an open call for tissue release, and we simply now just need a critical mass of funding and good proposals for the right kinds of genomic analyses to be carried out through people who have the means to do so.
Michael, maybe I can just comment a little bit. And Matt and I were having this discussion. I mean,
okay.
Of course, I think everyone in this room agrees it's a question of funding and where the money can come from. Obviously, with NCI sponsoring both an adult and soon a pediatric match and an enormous commitment there, the question is where can we, I think everyone in pharma would say, boy, having better characterized, genetically characterized tumors would give us much more meat on this bone. And it's a question, Matt and I were just trying to figure out what's the absolute dollar amount given the bank that's already there that we could actually do this. But it's really comes down to a question of
with the lower cost of sequencing now, this is going to be nothing like a TCGA effort.
I think that's what we need. We were just trying to do a calculation and figure it out.
5,000 canine tumors.
Yeah, we figured about $1 million. Yeah, we figured about $1 million. So,
But, I think it's also an opportunity to really create. As a community, there's a lot of individual efforts to do this and that, and the data end up in pockets that are not shared. And so I think it's our opportunity. Thank you. I'm Jaime Modiano from University of Minnesota. I also happen to be the president of CCOGC right now, not by popular election, but because somebody had to do it, and I was the only one standing after everybody ran. I'm a slow runner, and so, but anyway, I think this is really a great opportunity because we have the tissues, and we have removed, or we have tried to remove most, if not all, of the obstacles for these tissues to be released. Really, cost has not seemed to be a problem in our experience. There were other obstacles that we've tried to address. But I think that having these tissues go out in small batches to do small studies where the data remain locked behind a key in somebody's computer for months or for years, while it's very good for that investigator and ultimately, hopefully, the data will be published. One of the things that we have been discussing as a group is really the opportunity to partner with people where we say, we have these tissues. We would love to make them available. How do we do that without breaking the bank? How do we do that without having to close our door so that we can replenish these tissues and get more and more and more stuff for the things that are coming down the pike 20 years from now, while at the same time creating the data that we need. And so, we have paraffin blocks that can be used for tissue microarrays paired with the same samples that could be used for genomic profiling, either out of paraffin or fresh tissue. And so there really are great opportunities. And I would urge the people in this room to think about this and talk to your friend, neighbor, and pharma CEO who has the opportunity to really make these decisions. And just like we saw pharmas coming together and saying, hey, let's combine drug 1 with drug 2 and see if that works better. Perhaps we could get a lot of these big places and little places to work together and say, this would be a really good initiative to make into a public domain that we can all tap into. And I think that really is the solution. It's just a question of how do we get there.
Thank you. Let's come over here.
Hi, I just want to respond to the discussion. Our group is working with the CCOGC, as I mentioned, we've done the sort of profiling for both B-cell and T-cell lymphomas. And we have other analyses in progress for melanoma and osteosarcoma, and I believe that TGen is also doing work on melanoma. And of course, we would really welcome a large-scale effort. I think it really is what is needed in order to see exactly which dog cancers are translatable in what way. But speaking of the question of cost is a huge issue. And while the cost of sequencing has gone down, when it comes to exome sequencing, which is what we're doing because whole genome sequencing is too expensive, then the sequencing part actually becomes a relatively small portion of the cost. There's a huge portion that are the bait costs for actually doing the capture, and then analysis. As sequencing costs goes down, people become the expensive part of this work.
So Len, you've been waiting a long time.
Yeah, I sure have. Well, I
Why
was
don't you
trying to
go
be a
ahead?
gentleman, so occasionally. Len Lichtenfeld, American Cancer Society, and I've been standing here for a long time, so I don't know if my comments are still relevant, but I didn't want Lou to be out there by himself. As someone who's been very involved in the oncology world, I will share, I think that the question was asked, how much do people really know about this? And I have to say I agree with Lou. I was actually pleased to see that there was a slide up there today that we funded something back in 1980. I don't know what our funding stream is today, nor am I offering funding because I have colleagues who may be watching this who would be upset if I did. But I will be talking tomorrow, and I don't want to say the things I'll be saying tomorrow. But as someone who had literally was naive to this discussion, and I suspect there may be a couple of folks in the room who were, it's really been pretty enlightening. And as someone who, and I'll talk more about tomorrow, who's actually been personally involved with a dog who developed cancer, there are a lot of things that have to happen to make this work. And one of the things happens to be the number of veterinary oncologists who are around. I mean, there's some terrific people here who have done terrific work that I admire, but it is not generally accessible. But the opportunities, I think, are real. I mean, we've heard comments pro and con today, both from human drug development and what can be accomplished, but you also see clues of other things that are out there. Mucosal melanoma comes to mind because that's a spontaneously developing cancer. To the genomic issue, I think it's critical that be done. Because the first question that was asked by my wife, who happens to be an OB-GYN, I will say, and is not involved, and she better not be watching this when I say it. First question she asked is, well, these dogs are pedigreed. And there is a law for that, and no one's mentioned that along the way, because someone did allude to it a little bit today. The pedigree issue is, in fact, a real issue in the dog world, as I've known from a non-cancer related experience, but you know who did what to who along the way, and those records go back for some period. You understand what I'm saying. I don't know the nice way to say it in the canine talk. But the point being, the pedigrees are out there, but number two, the question about whether there's specific genetic markers and that have actually contributed to those cancers that are not relevant to human cancers that may have been passed on is certainly a question. But that's only one among many. But I think that what I've heard today, and actually we'll share one final thought. After having been asked to be involved in this discussion, all of a sudden you're watching TV, and you do have folks out there who are promoting canine cancer and the understanding the disease and the treatment. They're on popular television. If I'm seeing those ads, there are others. So the interest level will likely increase, and you're doing incredible work to try to coordinate and collaborate, but the need is probably going to increase as well. So blue sky, I don't know if that's the right word, but opportunities are likely going to be out there, and particularly when it comes to the correlative research that we've been discussing today, I think it's critically important. So thank you.
Thank you, Len. I'm not sure there was a question there, but I think we agree. We've got time for one last one.
Hi, I just want to note that actually I've just funded by NCI to do canine human comparative sequencing work. And this is actually on the GI cancer. I'm trying to get the money to do the mammary cancer also, but the data will be like a 10x whole genome sequencing data plus RNA-seq. So for like 100 canine cases. So those data will be released to the community like immediately once we generated it and publish it. And so they think that I need a tumor. So if you guys have any K-line GI tumors, just let me know, and we will sequence it, and we will share the data with you.
All right. Thank you for that. And on that note, we'll close session 5. And thank you all for a very productive day.